Good day everyone, welcome to the week 4 of the course Biological Data Analysis and Visualization with R. In the past 3 weeks we have learned about the basics of R. We have used some of the packages for statistical analysis as well as data visualization. We are now ready to move to the next step where we will be focusing on specific problem statement and we will be taking a data set and we will be analyzing that for the whole week. Okay. So, the agenda for this week is gene expression analysis and co-expression network. So, in this class I will first introduce gene expression data and then I will be talking about co-expression analysis, and from subsequent classes we will be talking about data sets, and over 4 classes we will actually do this analysis step by step hands on. Okay, so this is the agenda. So, I will first describe what gene expression data is. So, some of you might be familiar with gene expression data, but we will discuss the basics of gene expression data so that everyone is on the same page, and then we will be talking about co-expression network analysis. I will just very briefly introduce what is co-expression network analysis. Of course, we will have a much more detailed discussion in the next over the next few classes. Okay, so these are the keywords you will see. For example, next generation sequencing is the first keyword, because most of the gene expression data that we will be handling will come from next generation sequencing platforms like Illumina. And the keyword, the co-expression network analysis that we will see is called weighted gene co-expression network analysis, or WGCNA. So, let us now start with gene expression data, very brief introduction. So, we get gene expression data from microarray experiments. Earlier it used to be microarray experiments, but currently it is mostly next generation sequencing data. So, next generation sequencing platforms, they generate huge amount of data. So, what is gene expression data? So, we have samples, and for each sample we get expression values of all genes. Okay. So, earlier it used to be done with the microarray experiments where there was probe hybridization, and currently it is done with next generation sequencing platforms where the transcripts are sequenced. Okay. And the protocol roughly looks like this, very coarse grained, right. This is the protocol. So, what happens is that from samples there is total RNA extraction, then there is a step for mRNA enrichment, because we want to measure expression levels of genes, right. So, we are interested in mRNA only, right. So, in that case there is an mRNA enrichment, and this is also accompanied by ribosomal RNA depletion, right, because in cells most of the RNA that is produced is ribosomal RNA. So, we need to get rid of ribosomal RNA before we can actually get the signal for mRNA. Now, once this is done, the next step is cDNA synthesis, again depending on the platform. So, for example, in some of the NGS platforms you do not need to do cDNA synthesis, you can do direct RNA sequencing. But anyway, that is we are not going to discuss all these platforms here. And then once this is done, we can go for either library preparation and do the microarray experiments. That was the norm in earlier days, and currently it is we go through the library preparation and the next generation sequencing application. Okay. So, once we get this next generation, we do the next generation sequencing, and what we get is the read data which we can process to get a measure of expression of all genes in the sample. Okay. So, this is bulk RNA sequencing data, but we can also work with single cell RNA sequencing data, where instead of the full sample, right, where you have multiple cells, you get expression data of all genes in individual cells. Okay. So, this is something that is also possible nowadays, and you can we sometimes also work with single cell data. Anyway, so there are a lot of analysis that you can do. So, why are we interested in this gene expression analysis, or we measure gene expression for different types of analysis. So, one of the first analysis is the differential gene expression analysis, or DGE. Right. This is a very common form of analysis that many researchers are interested in. So, what is differential gene expression analysis? Very briefly is, for example, we are measuring expression levels of genes, all genes in 2 different conditions, for example, or we are measuring gene expression of samples from different cell types, right. So, for example, you have, let us say, normal cell types versus disease cells. Okay. So, we have tissues that come from healthy individual and one from diseased individual, and we want to compare, right, how the gene expression is changing based depending on the treatment or depending on cell type or depending on whether there is a disease or not. So, for that we do differential gene expression analysis, and this is a very common analysis that we apply. The second type of analysis that you can also do is we want to identify isoforms and alternative splicing events. So, as you are aware, right, in higher eukaryotes we have alternative splicing, and there are different isoforms arising from the same gene, and sometimes these isoforms have different functional roles in the cell. So, in those cases we want to study isoform distribution or quantify isoform expressions, and we kind of then can associate this with different cellular outcomes or differentiation, etc. We can also study non-coding RNAs. So, for example, we also do something called small RNA sequencing that may not be part of the mRNA sequencing that I just showed you. We are also interested in how non-coding RNAs change in their abundance, and then how they actually influence gene expression and cellular processes. So, differential gene expression analysis we can actually study. There are actually R packages to do this specifically. This is a very elaborate process. Again, this is a multi-step process that takes a quite a bit of time, right? Multiple statistical analysis that you have to do. There are a lot of things that are important in there. So, we usually start with the data. There is pre-processing. So, this pre-processing mostly involves, for example, normalization. This is one of the major steps in there, followed by identification of differentially expressed genes, and then followed by functional enrichment, which functional class of genes are showing differential expression, those kind of things. There are a lot of statistical tests involved, there are a lot of steps involved. This is something we have discussed in an earlier course, so we will not be discussing here. If you are interested, please feel free to look up this course. What we will be discussing in this class is something called weighted gene co-expression network analysis. Okay. So, what is this weighted gene co-expression network analysis, or WGCNA? Okay. So, here we start with gene expression data, and what we are interested in is looking at correlation pattern between genes. Okay. So, instead of differential expression analysis, etc., what we look into first is correlation pattern between genes, and we usually calculate correlation values between pairs of genes, right, and we have to do this for all pairs that are in our data set. Okay. So, that is one of the first steps, right. Now, the idea behind this is these genes that show correlation, very high expression correlation across different conditions, they are likely to be genes that are important, right, functionally important. Okay. So, when you do look into these correlation patterns, we want to identify these clusters or modules of genes that show high expression correlation. Okay. That is the idea, and these can actually have a lot of applications, right. There might have some specific roles in specific cellular processes. So, here is the idea, right, that is actually kind of introduced this idea, right, where looking into the signaling networks using and then found this as a target. Okay. So, what are the applications of this WGCNA analysis? Okay. So, the idea is, if the genes that are correlated, they might be members of protein complexes, right. So, we might able to identify these members, right, perhaps from our analysis, or maybe functioning in the same cellular pathway, right. So, that is why they are expressed in similar manner, right, because for example, there is some sort of metabolic pathway that is important for some process. So, this is something that might tell us, right, these are members of the same cellular pathways, and which ultimately means these genes are also likely to be regulated together, right. So, it also might have a common regulator, or maybe the genes that one of the genes that is within this cluster, right, that show high correlation could be a regulator of the other genes, right. So, as its expression, as the expression level of this gene, the regulator gene or the transcription factor goes up, it also induces other genes that are around it, right. So, it kind of also can give us some insights into gene regulation and also signaling, right. So, we can actually understand what are the genes that are regulated together, and we can then start identifying the regulator or the transcription factor that is regulating all these processes. Now, the most interesting part of this is once we have these clusters or modules, we can then look at this network, right, and we can actually do a lot of network analysis and try to identify what we call as hub genes. Okay. So, what are these hub genes? So, hub genes are genes that actually are likely to share a lot of connection with other members of that module. Okay. So, these are the genes that are highly connected, right. Highly connected means they show a high correlation with a lot of the members of the gene, right, members of the cluster or the module that we identify, and in that respect this is likely to be a very important gene, right. That's what we can assume, because this gene is showing very high correlation with a lot of members of the module. So, this is likely to be a very important gene. So, these are called hub genes, and these hub genes might be transcription factors or could be anything else, but this could be actually the key regulators of this specific process, right, and this could be targeted in disease. Okay. So, this is where the importance of this WGCN analysis is, right. So, these gene members may be so these are the members of protein complexes or cellular pathways. As we said, we also get some understanding of gene regulation, and the final one is the hub gene. We can identify this hub gene in the network, highly connected genes that could be important regulators of many cellular processes or some cellular processes, and they could be the targets for diseases. Okay. So, I will just mention some of these applications very briefly. So, for example, as I have already introduced this paper, right. So, this is the molecular targets in cancer, I mentioned this. So, this is again an analysis of oncogenic signal networks, and they actually identified a gene as a molecular target, right. So, I am also giving you the link so that you can go and read this. The applications, for example, here that has also been applied for identifying of angiogenesis genes in cancer, right. Here again in a multiple myeloma, right, they have actually looked into this angiogenesis related genes, and you can actually identify these clusters and hub genes, etc. So, not just in diseases, but this could all this application, this analysis can be also be applied for different types of other studies, right. For example, here is an example of this analysis applied to study of genes, right, that are important for organismal development. So, this concept of WGCNA was applied in human and mouse early embryos, right, and from on single cell RNA sequencing data, which actually allowed to identify those genetic programs that are likely to be important, the modules that are important for development. Okay. So, as you can see, you have variety of applications, and finally, here is another example of this identification of gene modules in neurodegenerative disorders, right. So, for example, the paper here I am showing is for autism spectrum disorder, but this has this similar concept of WGCNA has been applied to, for example, Huntington disease and other diseases as well. So, this is a wide variety of application that is possible with WGCNA. I have just mentioned only few, but there are now many publications that actually use this technique and very widely used to actually understand regulation and modules, etc., which finally leads to identification of hub genes. So, now we will move into how we can do the WGCN analysis, right. So, I have introduced what is WGCN analysis. Now, we want to do this analysis ourselves, right. So, that is the idea, that is the goal of this week, right. So, I will first introduce the steps that we have to follow for WGCN analysis. So, here is the R package. So, it should be WGCNA, right, instead of WCGNA. So, this should be WGCNA package. This is the paper here, and the R package is here. Okay. So, this is the paper that actually describes this R package, right, and it kind of stepwise mentions what are the process, what will be the process for doing this WGCN analysis and what we need to do at each step. Here is the R package. We will have a look at this R package and its manual, and then of course we will apply this package for our hands-on analysis. So let us now very briefly look into the steps, right? What are the steps that you have to follow? These are the kind of overall idea, right, how WGCNA analysis works. And then of course when we go into the hands-on, we will be talking about the workflow, right. So the workflow will go into a lot more technical details, right, and which we will use for our analysis. So here are the steps. So the first step is calculating the correlation, right. So we have to calculate the pairwise correlation between expression levels of genes. Okay, this is the first step, and this is a quite a heavy step, computationally heavy step, because as you can imagine, each sample might have 4,000, 5,000 genes at least for like organisms like bacteria or yeast, but for higher organisms it might have 20,000, 30,000 transcript values or gene expression values, right, and we have to do this pairwise correlation for all of them, right. So this is a huge combination, huge number of combinations. Now once we do this pairwise correlation, the next step is the construction of the co-expression network, right. So what we get at the end of pairwise correlation is a correlation matrix, right, and from this correlation matrix we can identify modules, right. If we could visualize, of course this is a huge data, this is very difficult to visualize, but if we could visualize, we can identify subsets, right, that show very high correlation. So if you can imagine in your mind, right, we have this correlation matrix, right, and let us say we can color code this correlation matrix in blue red color, right, showing very high correlation, red meaning high correlation. You can see these clusters or modules, right, subsets within that big matrix that are showing very high correlation. Okay. So that is the idea, that we actually pick out those subsets, right, from that pairwise correlation, and we construct the co-expression network. Okay. Now of course this sounds very easy, but this is a pretty complex step. So how do we decide on the cutoff, right? How do we decide what should be the correlation cutoff that we should be using to actually get this subset, right? That's the most appropriate subset. Okay, so that's a quite complex analysis. We'll deal with this when we actually go into the hands-on part. Okay, so once we have this, right, we have so what we are identifying at the end of this is identification of these modules, right, that are showing very high expression correlation. Okay, and these modules are likely to be biologically interesting. Some of them might be actually biologically interesting. Now when we identify these modules, there is one part that's very important, is we try to go into something called soft thresholding, which we will discuss in a lot of details. So the idea is, how do we choose this threshold is by using this soft threshold. But what should be that soft threshold is that there is not a fixed threshold, but there is a threshold that we can determine from the network that we have constructed, right, this co-expression network. Okay. So this co-expression network, the idea is that we want to approach a scale free network, and we have to pick the soft threshold value in such a way that we get a scale free network. Okay. So we will do this in the hands-on, we will see this in much more detail as we go towards the hands-on. Okay. So once we identify all the modules, some of them are likely to be interesting modules, right. So as I said, let us say we are dealing with healthy versus disease data, and we have taken all data and we have constructed this co-expression network, right, and we have identified some of the modules that show very high expression correlation. What we want to see is like how this correlation, right, is varying, or the score is varying between different samples, right, especially between healthy and disease sample, right. These are the biologically interesting modules, right. So if you are analyzing, let us say healthy versus disease samples, we want to pick those modules that show different behavior in healthy and disease samples. Okay. So those we can identify. Once we have identified the modules, we can actually identify those biologically interesting modules, and we can actually use other data sets as well or other criteria to actually pick out these interesting modules, right. So there are a lot of possibilities. I just mentioned one possibility, which is like there is different behavior of the module in different sample types, but you can superimpose other types of data and perhaps find out different biological interesting modules. So that's possible. And then once you do that, as I said, once you have these modules, you want to look at the genes of these modules, right? What are the genes in there, and do some network analysis, which might give us an idea about the hub genes. Okay. So these hub genes, as I said, are likely to be drivers of these processes, whether you have something like the disease, right, that we have seen, right. So we have, for example, we have seen some of the papers where they are looking at these hub genes in diseases, right. Those could be good biomarkers, those could be good therapeutic targets, or you can identify genes that are, for example, important for a developmental process or maybe neurodegenerative disorders. Okay, so here are the references for that I have introduced. So I just point out few references here, right. Some of the references I just mentioned, right. So this is the first paper that actually talked about this idea, WGCNA, and then here is the following paper that actually talks about geometric interpretation, right, how this gene co-expression network analysis works and what is the idea behind this. So if you are interested in the details of how this method works and how do you pick out these modules, etc., please go through this. And then of course you have different papers that we just mentioned, some of the applications of this method, and then the final one is for the R package. So to conclude, right, we have seen, right, overview of gene expression data, how gene expression data is generated. This is usually done by microarray and next generation sequencing. So this is something that we have discussed in an earlier course. So I have not gone over them. So if you are interested, please feel free to check that course. We have talked about some of the applications of some of the analysis that you can do with gene expression data. For example, differential gene expression analysis or isoform identification. So again, this differential gene expression analysis is a very popular method of analysis, and this is something that we have covered in an earlier course. There are R packages like DSEC2, EDGE-R, etc., that we had used in that course. So please head over to that if you are interested. The analysis that we will be focusing on, that we have talked about in this class and that we will be focusing in the subsequent classes, is the weighted gene correlation network analysis, or WGCNA. And I just briefly introduced the idea of WGCNA. So the idea of WGCNA is identifying the modules of highly correlated genes, because this might give us insights into gene regulation, or it will enable us to identify hub genes, right, that are probably driving a disease or maybe driving cellular processes, and at the same time some of these genes could also be targets for therapeutic treatment. We have talked about, I have just very briefly mentioned some of the applications of WGCNA, right. Of course, there are many such papers now showing the applications of WGCNA method for different purposes. So this method is very popular. And lastly, I talked about WGCNA steps, right. Very briefly, I gave you an overview of the steps that we follow for WGCNA analysis, and then as I said, right, we'll introduce these steps in much more detail when you go into the hands-on. That's the agenda for the next few classes. So we'll start with we'll first look into the WGCNA package in the next class. We'll introduce some data set, and then we'll start working on that. We'll follow all these steps to actually identify these interesting modules. So that will be from the next class. Thank you.